Hello, everyone, and thanks for joining us today. My name is Mason Brooks, and welcome to this installment of the Active Motif webinar series. Today, we'll be hearing from Dr. Felizza Gunderson, who is Manager of Epigenetic Services here at Active Motif. Felizza will be covering the popular techniques of ATAC-Seq and single-cell ATAC-Seq, and how those techniques can be applied to epigenetics research.
Fantastic. Thank you so much, Mason, for the introduction, and thank you all for dialing in. Like Mason mentioned, today we'll be covering single-cell ATAC-Seq, how it works, and what kind of experimental questions it can address. To start things off, I'd like to give you a brief agenda of what we'll be discussing today and what I hope you will gain by the end of this webinar. By the end of the webinar, you should know what is ATAC-Seq and what type of information it can provide, understand some of the limitations to standard ATAC-Seq, recognize how single-cell ATAC-Seq works, including knowing the similarities and differences in data representation between single-cell ATAC-Seq and ATAC-Seq, identify experimental questions that single-cell ATAC-Seq can answer. Furthermore, understanding what type of challenges one can encounter when performing single-cell ATAC-Seq experiments. And lastly, I'd like to leave you with some information regarding Active Motif's single-cell ATAC-Seq service. So to kick things off, what is ATAC-Seq? ATAC-Seq is an acronym for Assay for Transposase Accessible Chromatin. This assay allows for the interrogation of open and accessible regions of the genome. This is accomplished by the use of a TN5 transposase. Below is a schematic of the workflow for ATAC-Seq. ATAC-Seq starts by isolating nuclei from about 500 to 100,000 cells, and it's incubated with TN5 that is loaded with sequencing adapters. The TN5 transposase targets these areas of open chromatin and inserts a portion of this sequencing adapter. Once the fragmentation is complete, the fragmented DNA is then purified and followed by PCR to complete the sequencing adapters and introduce indices that are used for multiplexing for downstream next-generation sequencing. For additional information on ATAC-Seq, including tips and tricks, stay tuned for an upcoming webinar from the Active Motif team about all the ins and outs to ATAC-Seq. What kind of information can examining open chromatin provide? Some examples include it allows you to quickly determine if epigenetics is involved in a response. If you don't have a candidate histone modification or a known transcription factor to examine, it is the logical choice to get a lay of the epigenetic landscape. From ATAC-Seq, you can also infer which histone modification or modifications are of interest based on correlation of open chromatin regions to genomic annotations. One can further analyze these areas of open chromatin by using motif analysis to identify what transcription factors may be driving cell fate, disease, or response. And lastly, you can gain mechanistic insight into how your pathway, your cellular response, or disease is regulated and give you the next steps to identify your next set of experiments. What are some potential limitations to ATAC-Seq or standard ATAC-Seq? Like I mentioned, ATAC-Seq is a great tool that allows researchers to uncover areas of open chromatin and gain that mechanistic insight. That being said, like any method, there are some limitations to keep in mind. One point to consider with standard ATAC-Seq is that ATAC-Seq averages out cellular heterogeneity. For example, here we have a big weak track in which we're examining areas of open chromatin at the gene CD33. These peaks of open chromatin are in blue. If you don't know what a big weak track is, we are having an upcoming Bioinformatics 101 webinar presented by our Active Motif team, so please stay tuned if you wanted to learn all about bioinformatics and epigenetics. With standard ATAC-Seq, like I mentioned, it averages out cellular heterogeneity. So looking at this type of data, one won't be able to discern which subpopulation of cells are driving this particular response. Another point to consider is that there may be isolation strategies like FACS sorting or magnetic sorting that could alter the biology due to sample manipulation. There are times in which you would need to use FACS sorting to be able to isolate these rare populations, and this may have an impact on the biology. Given these points, if you are interested in understanding the epigenetic landscape of open chromatin in specific subpopulations or rare populations, single-cell methods may be able to help you. So what is single-cell ATAC-Seq? This is the same method that uses the TN5 transposase as in standard ATAC-Seq. However, it now combines that methodology with barcoding of individual cells, and this allows researchers to profile chromatin accessibility at single-cell resolution over thousands of nuclei. There have been several different single-cell ATAC-Seq methodologies that have been developed over the years. Some of the more popular include combinatorial cell indexing strategies or the use of microfluidic devices. And I'll go into some details and compare and contrast these two methods in the next slide. So here we have the key steps in the standard ATAC-Seq workflow. We have cell lysis, where we isolate the nuclei, transposition, amplification, and subsequent sequencing. With the combinatorial cell indexing method, you isolate nuclei and they're molecularly tagged in a 96-well plate format and then barcoded with transposase complexes. These tagged nuclei are then pooled and diluted and redistributed to a second 96-well plate at about 15 to 25 cells per well. Each well will then correspond to a pool. This is all done with fax sorting. After this is accomplished, PCR is used to finish the sequencing adapter, much like the standard ATAC-Seq, introduce a barcode or index to allow for multiplexing for next generation sequencing. The microfluidic method, on the other hand, still uses the same principles and key steps of the ATAC-Seq workflow. However, the transposition occurs at a nanoliter scale using a microfluidic device. Once that transposition event is completed, amplification is used to complete the adapter and allow for library construction for next generation sequencing. Now, both of these methods use very similar methodologies, still following the same steps of the standard ATAC-Seq workflow in combination with individual nuclei barcoding. Now, which method you choose to perform your single-cell ATAC-Seq, whether it's the cell indexing method, microfluidic method, or others, will really be dependent on what platform you have at your disposal. Over the recent years, there have been many companies that have emerged that utilize the microfluidic methodology for single-cell analysis. These include Fluidigm, Bio-Rad, Takara, and also 10x Genomics. I'm going to highlight 10x Genomics technology and how it works with single-cell ATAC-Seq. For the 10x Genomics workflow, we start off with isolated nuclei that is incubated with a transposase in bulk, as depicted here in this Eppendorf tube. These nuclei that have been incubated with the transposase are loaded onto the microfluidic device, which is called the 10x Chromium Controller, which is depicted here. And it also is incubated with these barcoded gel beads. These gel beads, if we take a closer look, are covered with thousands of different oligos, and each bead has a unique barcode, such that when a bead comes in contact with a nuclei in the microfluidic device within a capsule of oil, you will be retaining one bead to one nuclei. Having this unique barcode on each of the individual beads allows you to then trace back to be able to understand from what cell did the signal come. Once it goes through the chromium controller, you then now have these complexes of nuclei and beads. You then perform linear amplification to amplify the signal. You remove the oil, and then you pool these barcoded fragments to then proceed on with library preparation. So although single-cell ATAC-Seq does use those same principles of standard ATAC-Seq, there are some differences in the way single-cell ATAC-Seq data is represented compared to standard ATAC-Seq. Here again, I have an example of a big wig track looking at this particular gene loci, where the peaks of open chromatin are indicated in blue. Again, with standard ATAC-Seq, this is an average of all the signals. With single-cell ATAC, not only are you able to look at the average signal, You then have the data represented in what is considered a T-SNE plot, or now also a UMAP plot. In these plots, each color corresponds to a group of cells that all contain, or a group of nuclei rather, that all contain the same open chromatin profile. The T-SNE plots or the UMAP plots allow, because of the data composition of single cell, is highly dimensional, and this allows for a clear representation of each of the different cell clusters or clusters of nuclei that can be identified. One can further extrapolate from each one of these different clusters what the open chromatin profile looks like. Here on the right, I have the same gene that is represented here in the standard ATAC-Seq, but now we can see for each one of the 12 clusters that were identified with single-cell ATAC-Seq, you are now able to see what those individual peak profiles look like. Now, given the complexity of single-cell ATAC-Seq and the amount of data that single-cell ATAC-Seq generates, having this level of resolution can help many researchers better understand and classify different cell populations in a heterogeneous population or deconvolute tumor microenvironments. In the next slides, I'll outline how researchers are using single-cell ATAC-Seq and what type of experimental questions it can address. For example, some questions that single-cell ATAC-Seq can address include, like I mentioned earlier, classifying different cell populations that are within a heterogeneous population, help you understand what is comprising the tumor microenvironment, what cell populations are present, what populations are driving a drug response. It can aid in examining cell differentiation and cell trajectories during development. And it can also help identify and build gene regulatory networks involved in development or disease. One example in which single-cell ATAC-Seq can aid in these types of experimental questions is that it can allow for marker-free identification of different cell types. In this study, the authors were trying to understand how single-cell ATAC-Seq can be used to understand the chromatin landscape during human hematopoiesis. Human bone marrow and blood were collected and single-cell ATAC-Seq was performed. What the authors found is that they were able to identify 31 different cell types or cell clusters that corresponded anywhere from early progenitors to end-stage cell types. From the single-cell data, if you drill down a little deeper, they were able to identify a variety of different cis-regulatory elements that correspond to each one of the different cell types. So the enhancers and promoters associated with each one of the different cell types as represented in this heat map. The authors were also able to identify, when looking at plasma B-cell differentiation, how can you order cells along this trajectory from hematopoietic stem cell all the way up to a plasma B-cell. They were able to identify what transcription factors were involved at each step of this trajectory. Single-cell ATAC-Seq can also help deconvolute the tumor microenvironment. In this example, biopsies from patients with basal cell carcinoma receiving PD-1 immunotherapy were used to try to understand what the different cell types were comprised the tumor microenvironment. Single-cell deconvolution of the tumor microenvironment revealed distinct types of immune cells, stromal cells, and malignant cells, as depicted here in this UMAP plot. Interestingly enough, the authors noticed that stromal cells and immune cells all clustered together independent of the patient. However, when you look at the tumor cells, there were 4 distinct clusters that corresponded and were patient-specific. From this single-cell ATAC-Seq data, the authors can then look at the chromatin profile of different regions that are involved or genes that are involved in T-cell activation. In the tumor microenvironment, T cells can be activated by targeting inhibitory receptors on T cells and stromal cells. The differences in chromatin accessibility between these were examined, and they decided to look at 3 known inhibitory ligands, CD47, TGFB1, and PDL1. What the authors noticed is that there's differences in the open chromatin profile among the different sample types. There's differences in cis regulatory elements or open areas of chromatin at these regions between tumors and stromal cells. Also, open areas of chromatin that are specific to only the stromal cells, and you're able to discern differences even within each one of the patient samples and all of the other cells within the tumor microenvironment. So just to summarize how single-cell ATAC-Seq can aid in your research and what type of experimental questions it can address. Single-cell ATAC-Seq allows for the interrogation of open chromatin at single-cell resolution. Single-cell ATAC-Seq methodologies include combinatorial cell indexing or the use of microfluidics. Examining areas of open chromatin can provide mechanistic insight into how your pathway, cellular response, or disease is regulated. Single-cell ATAC-Seq can help identify what cell population or populations are driving disease or drug response. And lastly, single-cell ATAC-Seq can deconvolute heterogeneity in a mixed population, potentially eliminating the need of harsh isolation methods. Now, when adopting any new technology, optimization is key and is required for everybody's own specific system. During this optimization, some challenges may arise. Some challenges include lysis conditions. It may be very specific to your cell type or tissue type. Tissue dissociation may be a challenge for some and just something that's specific to certain types of tissue. If you don't have the availability of a FACS sorter, let's say, or availability of one of these microfluidic devices, there is the potential of a capital equipment investment. Furthermore, sequencing costs, because of the thousands of nuclei that are being sequenced, sequencing costs may be inhibitory. And also the downstream bioinformatic analysis may be challenging for some. If you don't have the time, expertise, or resources to tackle these points to try to optimize and get single-cell ATAC-Seq working in your own lab, this is where Active Motif can help. Active Motif does offer a full end-to-end single-cell ATAC-Seq service. Here I have a brief outline of what's included in our single-cell ATAC-Seq service. We find the best lysis conditions for your sample by performing first a sample validation to make sure that we have all of the appropriate conditions to be able to deliver high quality data to you and before embarking on the full-scale experiment. If the sample validation is successful, then we proceed with the full-scale single-cell ATAC-Seq service. We then incubate the nuclei with the transposase enzyme. We then use the 10x Genomics Chromium controller and reagents to barcode individual nuclei. We aim for typically about 3,000 to 7,000 nuclei. The nuclei are pooled for library preparation, and each pool will contain a common library index to allow for multiplexing on the sequencer. With all of our services, including our single-cell ATAC-Seq, all pre-sequencing QC checks are included, including library qualification to ensure that the library is of the appropriate size, and also quantification of the library. Sequencing is also included with our full-scale single-cell ATAC-Seq service. We sequence to a depth of 250 million reads, which roughly equates to about 50,000 reads per nuclei. And also all the downstream bioinformatics is included with our full-scale single-cell ATAC-Seq service. This includes analysis of the single-cell data using the 10x Genomics Cell Ranger software and all the subsequent deliverables. However, in addition to that, we then take that single-cell ATAC-Seq data and run it through Active Motif's own ATAC-Seq analysis pipeline to generate pseudo bulk ATAC-Seq data. So you will have pseudo bulk ATAC-Seq data at the end and also single-cell ATAC-Seq data. To go into a little bit more of the specifics of these data deliverables for this service, for the single-cell ATAC-Seq service, the deliverables include all raw sequencing files, which are the FASTQ files, BAM alignment files, all the Cell Ranger raw analysis files, a HTML run summary file that gives you some high-level overview statistics of the single-cell attack-seq run, and all the files that are required for visualization of those T-SNE plots, those clusters of different colored, the clusters of cells, as well as the peak profiles for each of the individual cell clusters. All of those files are included. In addition to that, you will be receiving Active Motif's bulk ATAC-Seq full analysis deliverables, which include those visualization files, which are those peak profiles that I have been showing throughout this presentation, as well as differential analysis. And again, if any of this seems foreign to you, I highly recommend tuning into our upcoming Bioinformatics 101 webinar. And so before I let everyone go, I wanted to leave you guys with some resources. If you wanted to learn more about ATAC-Seq, single-cell attack, what are the differences, what Active Motif might be able to provide, I'm leaving you with some links to our Active ATAC-Seq information, as well as additional resources to single-cell attack. Also, please stay tuned. Check your email for upcoming webinars, including ATAC-Seq and bioinformatics. And with that, I wanted to thank everyone again for dialing in. I hope you found this webinar useful. And for additional information, feel free to visit our website at activemotif.com or contact us at techservice@activemotif.com. Have a wonderful day.